Less than a dozen Denisovan fossils exist, yet their DNA transformed our understanding of human evolution. In 2010, Krause, Paabo, and Reich sequenced a finger bone from Denisova Cave, revealing a distinct lineage with astonishingly low genetic diversity and a population size never exceeding 10,000. Meyer's high-coverage genome later confirmed the Denisovans' extreme demographic fragility and fragmented geography. Despite interbreeding, most Denisovan DNA vanished through selection and absorption. So why did an entire lineage, spanning 170,000 years, almost disappear without a trace? The answer lies in the convergence of genetic, ecological, and demographic forces now uncovered by DNA analysis. Denisovan fossils are exceptionally rare. Fewer than 12 bone and tooth fragments have been confirmed, scattered across a range that stretches from southern Siberia to Southeast Asia. The most significant site is Denisova Cave in the Altai Mountains, where a finger bone and several molars were uncovered from layers dated between 287,000 and 50,000 years ago. In 2019, a robust mandible from Baishia Karst Cave on the Tibetan Plateau was identified as Denisovan, pushing their known presence to at least 160,000 years ago and over 3,000 meters above sea level. A single molar from Tam Ngu Hao 2 Cave in Laos, dated to roughly 120,000 years ago, extends their reach deep into Southeast Asia. Despite this immense geographic and temporal span, the physical record remains fragmentary. No complete skeletons, no cranial vaults, only isolated teeth and bone fragments. Most sites yield only a single specimen, with no evidence for sustained occupation or population density. The scarcity of fossils and the absence of diagnostic tools or art make it impossible to reconstruct Denisovan behavior or appearance from bones alone. This profound lack of material evidence left major questions about Denisovan biology and history unresolved until the arrival of ancient DNA analysis. In 2010, Johannes Krause, Svante Paabo, and David Reich led the effort to extract DNA from a tiny finger bone found deep in Denisova Cave. Using advanced sequencing methods, the team achieved coverage exceeding 5,000-fold for the mitochondrial genome, an unprecedented depth for ancient DNA at the time. The results showed that Denisovan mitochondrial DNA split from the line, leading to both Neanderthals and modern humans nearly 1 million years ago. This divergence was almost twice as great as the difference between Neanderthal and modern human mitochondrial genomes. Now, the same mitochondrial sequence was later found in a molar from the cave, confirming the signal came from a single hominin group, not from contamination or mixed remains. The mitochondrial data alone established Denisovans as a distinct lineage, not a variant of Neanderthal or early modern human. These findings provided the first concrete evidence that a previously unknown branch of archaic humans had survived in Asia until at least 50,000 years ago. The precision of the sequencing, with coverage exceeding 5,000-fold and contamination kept below 1%, set a new standard for ancient DNA research. This mitochondrial breakthrough set the stage for sequencing the nuclear genome, which would soon reveal even deeper insights into Denisovan biology and their relationship to other hominins. Matthias Meyer and the Max Planck Institute team achieved a milestone in 2012 by sequencing the Denisovan nuclear genome to an average depth of 30 times, covering 99.9% of the mappable human genome. Contamination estimates were kept below 0.5%, ensuring the reliability of the data. The genome revealed an exceptionally low heterozygosity of 0.022%, about one-fifth the value found in present-day Africans. This low level of genetic diversity suggests that Denisovans lived in small, isolated populations for tens of thousands of years. Population size reconstructions, using the PSMC method and allele frequency spectra, consistently place the effective population size, or NE, between 1,000 and 10,000 individuals over the long term. Analyses of runs of homozygosity found only short tracks, which rules out recent inbreeding as the cause of low diversity. Instead, the data point to a metapopulation structure, with Denisovans scattered in fragmented groups that rarely exchanged genes. This chronic subdivision, rather than a single bottleneck or close-kin mating, underpinned the demographic fragility of the Denisovan lineage. The genetic evidence from the high-coverage genome provides a quantitative basis for understanding why Denisovans were so vulnerable to extinction pressures across their vast but sparsely populated range. Denisovan fossils define a range that spans from the frigid Altai Mountains to the humid forests of Southeast Asia, but each discovery stands alone, separated by thousands of kilometers and years. The Shahe mandible, recovered from Baishia Karst Cave on the Tibetan Plateau, is dated to approximately 160,000 years ago and found at an altitude exceeding 3,000 meters. Its robust structure and dental traits match the genetic profile established from Denisova Cave, confirming a Denisovan presence in extreme high-altitude environments long before modern humans arrived. In Southeast Asia, a single molar from Tam Ngu Hao 2 Cave in Laos, dated to around 120,000 years ago, extends the Denisovan record deep into the tropics. The physical distance between Xiahe and Tam Ngu Hao 2 exceeds 2,500 kilometers, yet no intermediate sites have yielded comparable fossils or genetic material. Each find is isolated in both geography and time, with no evidence for continuous occupation or migration corridors. The high-altitude Xiahe mandible and the tropical Tam Ngu Hao 2 molar demonstrate that Denisovans adapted to a broad spectrum of environments, but the fossil record reveals only scattered, disconnected populations. This geographic fragmentation, visible in the archaeological record, sets the stage for the genetic complexity uncovered by admixture studies and explains why Denisovan ancestry in modern populations displays such a patchwork pattern. Catherine Browning and colleagues applied a reference-free statistical method called SPRIME to more than 5,600 modern human genomes, and they uncovered 2 genetically distinct Denisovan admixture events. The first, pulse, closely related to the Altai Denisovan genome, contributed the majority of Denisovan ancestry found in present-day Papuans and other Melanesians, who retain between 4 and 6% of their genomes from this archaic source. The second, more divergent Denisovan component appears in East Asian and Southeast Asian populations, and it is not closely matched by the Altai genome, indicating gene flow from a different Denisovan group. This 2-pulse model shows that Denisovan populations were not homogeneous, but instead formed a patchwork of genetically distinct groups, each contributing uniquely to the ancestry of modern humans in different regions. The SPRIME approach did not rely on direct comparison to the Altai genome, which allowed detection of archaic segments from unsampled Denisovan lineages. Subsequent replication using D statistics and F4 ratio tests confirmed that the 2 Denisovan components are genuine and not artifacts of reference bias. The divergent nature of the second pulse implies that Denisovans themselves were deeply structured, with long-term isolation between Eastern and Western populations. These findings explain why Papuans and Melanesians carry much higher Denisovan ancestry than East Asians, and why the Denisovan genetic legacy in Asia is so fragmented. The admixture pulses predate the first successful settlement of Sahel, indicating that Denisovans interacted with modern humans before their expansion into Australia and New Guinea. Ancient genome sequencing has uncovered traces of Denisovan ancestry in East Asia, but at much lower levels than in Oceania. The Tianyuan individual, a modern human who lived near Beijing around 40,000 years ago, carries clear segments of Denisovan DNA. Another specimen, the Salkit skullcap from Mongolia, dated to 34,000 years ago, also shows Denisovan introgression. Both genomes demonstrate that Denisovan genes were present in the region long after Denisovan fossils disappeared from the archaeological record. Present-day Han Chinese, Japanese, and Korean populations carry about 0.2 to 0.4% Denisovan ancestry, a proportion so small it can only be detected using high-resolution statistical methods. This low-level signal suggests that Denisovans did not persist as a separate population in East Asia, but were absorbed into expanding groups of modern humans. The DNA segments inherited from Denisovans in these populations are highly fragmented, reflecting thousands of generations of recombination and the action of purifying selection. No evidence supports ongoing coexistence or hybrid zones after the initial admixture events. Instead, the genetic legacy of Denisovans in East Asia is best explained by demographic absorption. As modern human populations grew, Denisovan lineages were gradually diluted until only faint traces remained. The genomes of Tianyuan and Salkit serve as temporal markers, capturing the last genetic echoes of Denisovans in northern Asia before their distinct identity was erased from the fossil and genetic record. After Denisovan DNA entered the genomes of modern humans, most of it did not persist. Comparative analyses by Sankararaman and Vernot in 2016 mapped the distribution of Denisovan segments in over 5,000 contemporary genomes and found that only 1 to 3% of the original Denisovan genetic material remains today. The rest has been systematically eliminated by purifying selection, an ongoing process that removes deleterious archaic variants from the gene pool. This filtering is not random. Large regions of the genome, known as deserts, spanning more than 10 megabases, are almost entirely devoid of Denisovan sequences. Hawks and Akey, in a 2024 study, refined the boundaries of these deserts and showed they overlap with areas of high functional constraint, such as genes essential for development or cell maintenance. These regions are especially intolerant of disruptive archaic alleles. The pattern is clear. While admixture initially introduced hundreds of millions of base pairs of Denisovan DNA, the vast majority was gradually purged, leaving only a small residual fraction scattered across the genomes of present-day Asians, Melanesians, and indigenous Americans. This deficit sets the baseline for understanding the rare Denisovan variants that did survive, those that proved beneficial enough to overcome the relentless force of genetic filtering. A handful of Denisovan genetic variants survived the broad sweep of purifying selection because they provided immediate advantages to modern humans. The best-known example is the EPAS1 allele, a segment of DNA inherited from Denisovans that now appears in up to 90% of Tibetans. EPAS1 regulates the body's response to low oxygen, conferring a clear benefit for life above 4,000 meters on the Tibetan Plateau. Its rapid rise in frequency, traced by Huerta Sanchez and colleagues in 2014, is consistent with a strong selective sweep, with a selection coefficient estimated at roughly 3%. Another Denisovan-derived gene, MUC19, reached fixation in many indigenous American populations. MUC19 encodes a mucin protein important for respiratory immunity, and its high frequency is likely the result of pathogen-driven selection after the first migrations into the Americas. In Oceania, segments of the HLA-A and HLA-B immune loci show clear Denisovan ancestry. These alleles contribute to antigen presentation and are unusually diverse in Papuans and other Oceanian groups compared to global averages. The persistence of these adaptive variants demonstrates that, while most Denisovan DNA was eliminated, a few key regions conferred enough of a survival advantage to be retained in modern populations. Stratigraphic analysis of Denisova Cave reveals alternating layers containing Denisovans and Neanderthal remains, indicating both groups used the site over several thousand years. In 2018, Vivian Sloan and colleagues sequenced the genome of Denisova 11, a bone fragment from a 12-year-old female. The results showed she was a first-generation hybrid, with half her genome inherited from a Denisovan father and half from a Neanderthal mother. This direct evidence of interbreeding confirms that the two groups overlapped not just in time but in physical space. Demographic modeling by Hubis and collaborators in 2021 used joint site frequency spectra to estimate that Neanderthal populations maintained an effective population size 5 to 10 times larger than Denisovans throughout the late Pleistocene. While Denisovan effective population size ranged from 1,000 to 10,000, Neanderthal effective population size was consistently between 5,000 and 50,000. Larger, more stable Neanderthal populations would have enjoyed a demographic advantage, increasing their resilience to environmental shifts and resource competition. The genetic and stratigraphic evidence demonstrates that Denisovans were already declining before the arrival of modern humans, pressured by both their own fragmentation and the competitive presence of more numerous Neanderthal groups. The Denisova 11 hybrid stands as a rare genomic record of this contact, but the population size imbalance set the stage for further Denisovan decline. Population genetic simulations led by Browning and Hubes demonstrate how demographic swamping erased Denisovan identity. Models parameterized with a Denisovan effective population size between 1,000 and 10,000 and a modern human effective population size exceeding 20,000 show that even low levels of sustained gene flow can dilute the genetic signature of a small group within a few thousand years. Sediment DNA profiles from Denisova Cave reinforce this pattern. Denisovan mitochondrial fragments are consistently detected in layers up to about 50,000 years ago, but they drop abruptly to background levels in younger sediments. This disappearance coincides with the first archaeological evidence of modern human presence at the site. Paleoclimate proxies indicate no significant environmental shift at this boundary, ruling out climate as the primary driver. The timing and trajectory of Denisovan DNA loss in both simulations and sediment records align, supporting a model in which expanding modern human populations absorbed and replaced the last fragmented Denisovan groups through repeated admixture rather than catastrophic extinction. Multiple forces acted together to erase Denisovans as a distinct lineage. Fragmented populations, each with a small effective size between 1,000 and 10,000, faced chronic isolation and limited gene flow, as established by Meyer's high-coverage genome and PSMC reconstructions. Neanderthal groups, with effective sizes up to 10 times larger, competed directly for territory and resources, as shown by joint site frequency modeling from Hubis and the stratigraphic overlap in Denisova Cave. When modern humans expanded across Asia, demographic swamping accelerated the loss of Denisovan identity. Continuous low-level admixture, modeled by Browning and supported by sediment DNA profiles, diluted Denisovan genomes to a residual signal. Purifying selection then removed most archaic DNA, except for a few adaptive variants. The extinction of Denisovans was not the result of a single event, but the convergence of fragmentation, competition, absorption, and selection acting in concert. Today, the Denisovan legacy persists in the genomes of hundreds of millions of people. Population scale surveys estimate that between 300 and 500 million individuals across Asia, Oceania, and the Americas carry detectable segments of Denisovan DNA. In Papuans and Melanesians, nearly everyone retains 4 to 6% Denisovan ancestry, while smaller fragments appear in East Asians, Indigenous Americans, and Highland Tibetans. These surviving archaic variants, such as EPAS1 for high-altitude adaptation and MUC19 for respiratory immunity, have become essential reference points for modern genomics. Researchers use Denisovan sequences to trace the history of human adaptation, disease resistance, and migration. The Denisovan genome, first sequenced from a finger bone in 2012, now serves as a critical comparative baseline in studies of ancient admixture and evolutionary medicine. This living inheritance, scattered but enduring, is the final genetic trace of a vanished lineage. Today, Denisovan DNA persists in the genomes of over 1 billion people, as documented by Reich, Browning, and Huerta Sanchez. Each adaptive variant, EPAS1, MUC19, and HLA alleles, marks a trace of ancient survival strategies. As genomic studies accelerate, the Denisovan story is no longer about extinction. Their legacy endures in our biology, reminding us that human evolution is still unfolding. Share your perspective in the comments below.